Okay, so today is the rust class, meaning the starter class by me. Okay, at the last class I introduced the how to use blast search to estimate the bacterial species by using the sequence data. Yes, so as compared with the database deposited in the NCBI, and so after using the research of blast, we can estimate bacterial species. Okay, but clearly, if we want to know the bacterial species or the organism, not only bacteria but also mammalian species, so it's better to use the phylogenetic analysis. Phylogenetic analysis. Okay, today just I'd like to introduce the how to construct the phylogenetic tree, and so how we can understand the bacteria or the organism the classification are based on the phylogenetic the analysis. Okay, so after that, so shortly I'd like to show the how to use some software to construct the phylogenetic trees, and so I will give them some the mini working about this field. Okay, so that's why so shortly I'd like to prepare the movie lectures, and then so just could you please follow with my procedures and prepare some the result. It is one of the mini report. Okay, let's start. Okay, let's start the today's class. Just shortly I'd like to show the how to construct the phylogenetic tree. Okay. Yeah, so after extraction of DNA, for example, in that case from bacterial isolate in a laboratory, we can get the sequence data like this. Could you please see this one? Yes, this is just a raw data about sequences. Yes, so in that case, to identify, the bacteria we focused on the 16 srDNA, and after PCR, we try to get the sequence data from PCR products. Okay, so as you can see, here, so there's some the the numbers. Yes, from here, so the our sequence started, and from the 10, 20, 30, and so until 200, 300, 400, and then finally the until approximately 1,200 base pair, we can success to get the sequence data. So as you can see, you can see each peak, sharp peak, and with different colors, red or green and black, something like that. So this peak and color mean that they are nucleotide of DNA. So in other words, meaning A, T, G, or C. Yes, like this we can in order we can get the sequence data. But please focus on some the peak, the shape of peak like this. Yeah, so around here, so we cannot see the clear peak, and a little bit around here, so flat. But after that, we can see the confirm the clear peak. So we cannot use this one because this is not clear data. So we need to utilize the very very sharp peak around here. Okay. Okay, and so this is the first first half of sequence data, but please look at the latter half of the sequence data. Approximately after 1,000. Around here there are some sharp peaks, but here a little bit flat, and so sometimes overlapped between two peaks. And finally, the shape of the peak is not looks like irregular, so we cannot utilize around here. So it's better to just cut off until around here. Yes. Okay, so our PCR, so maybe you know already understand what is PCR, so how we can carry out the PCR, what is the mechanism of PCR, but to make it sure, shortly explained. Okay. Okay, so after extraction DNA from your samples, so meaning like microbes or fish, plants, mammalians, but anyway, so we can get the double strand DNA from organisms. And in process of PCR, so by heating, we can get the single strand DNA, this one and this one. Okay, so continuously we can, get the PCR products, but for it we need to utilize primer to read the sequence, and also, with support of enzyme, the polymerase, right? Okay, so in case of here, we can put the forward primers, and so based on the reaction polymerase, so primer can recognize the in each the sequence and we extend we can understand it. Okay, the case of the here, so the same, we can utilize reverse primers and try to extend based on their enzyme or polymerase reactions extension, and we can get the sequence. Okay, like this we can, continuously we can get the PCR products. But the important things, after the, finish our PCR, we can get the, full ranks of 16 sRDNAs. In that case, we need to, in my case, the for bacteria, so the ranks of the 16 sRDNA is approximately 1,600 base pairs. In that case, we can use two base pair, two primers like this, forward and reverse, respectively. Yes. So in the process of sequence, the forward primer should be, recognized and read the sequence information like from this way, like this, But the case of reverse primer is opposite. Yes, opposite. So it means, the when we get the full ranks of 16 DNA, so we have to take care of direction, and also, from here, if we look at the, from here to this sequence, so this sequence, the information should be opposite. In other words, complementary between two are the sequences. So it means, after getting the sequence by forward primer and reverse primer, so when we combine together with two sequence information, we need to take care about the reverse primer because the direction is opposite, and also, our sequence also are complement. So it means that we need to little bit edit before getting the full length of the 16S DNA. Okay, after that, so I explained how we can do it. Okay, as I explained, to get the almost the full length of the 16S RDNA, we need to use two primers. It means the first primers and the second primers. For example, in that case, here, so I isolated the strain FK1. About this strain, we need to get the two sequences by using forward primer. In that case, 27 F mean the forward primers. But another one is, here, so 104092 R mean the reverse primers. Okay, so by using two sequences, we can construct the full length of the 16 sRDNA. So let's try to open this data, the forward primers. Yes, as you can see here, yes, like this we can get the data. This is from 3 dash to 5 dash, like this. This order. So in that case, simply we can use the directly these sequences, and let's try to copy. And so in my case, just I used the what softwares. Let's try to paste here like this. Okay, and this is the information about the sequence by using forward primers. Okay, and so another one, so meaning the reverse primers. Let's try to open here. Okay, as same with the case of forward primer, we can get the sequence data like this. But as I said, the case of reverse primer, the direction of sequence is the opposite. Yes, so to construct the full length of the 16 srDNA, in case of the reverse primers, we need to make the complement. Complement is how we can construct the complementary sequence data. So there are so many methods, but in my case I use this one, BioEdit. Sorry, maybe you cannot see the information. Okay, here. Okay, this one. Yes, can you see it here? This is a software, one of the the edit the sequence data. The name is BioEdit Sequence Alignment Editors. So this is free software. So if you search in Google or Yahoo or something like that, you can get this software supplied by the BioEdit Institute. Yes. So let's try to open that file, reverse primers, the sequence data. Okay. Open. Okay, please wait. So many data. Okay, where is it? Please wait. Okay, so I found this one, the sequence the data from the reverse primer. Yes, so now already the data was output in this software BioEdit. And so we need to complementary sequence data about this sequence. So could you please, click from first the nucleotide and so final one. Could you please remember the last the alphabet GCT, and after that after getting their complement sequence data, the alphabet should be changed opposite because of the complement. So it means the alphabet should be changed to the AGC. Let's try do it. Okay. So how we can get the complementary sequence data? Just we can here, yeah, as you can see here, copy reverse complement. Why reverse? Complement means the opposite sequence data about the against the right. In case of T, so the complement means the A, and also we need to understand the direction because we are carrying that PCR forward and reverse like this. But in case of the reverse primer, we need to get the opposite way, opposite direction of the sequence data. Okay, so could you please push this the command? Yes, already the reverse complementary sequence data was copied in computers. So let's try to paste here. Yes. Okay, so please focus here. The first three alphabet A, G, C. So as I said, do you remember me that I said this original data about the sequence. Yes, so GCT. So let's try to make it the complement, and the direction should be changed. It means the reverse. So should be AGC. Yes, AGC. Yes, like this. So we can success the to get the reverse complement sequence data. Yes. AGCAA to, let's make it sure, AA, but in that case, TCG, TT, opposite complement mean the AA, yes, AA, it's correct, yes, successfully I could get the reverse complement sequence data. Okay, so it means, in case of 16 srDNA, this is start from the 1, 2, 3, 4, 5, something like that, these directions. And so from the 3 to 5, and the 3 to 5 like this. But this is there are some points overlap together in case of forward primer and reverse primer, because we need to combine together, Okay, so let's try to look for the overlap sequence, the area, and let's try to combine. Yes. So as I said, the first maybe 40 or 50 characters should be deleted, because the data of the sequence, the peaks, the shape of peak is very, very bad. Yes, as I said, meaning around here. Yes, around here, not so good. So in that case, it's better to cut off. Yeah, I cut off here. Okay. And then, so maybe good data, very sharp peak around here. Yes, but as I said, at the end of the sequence, for example, around here, not so good. Yes, so I think this is just case by case, but approximately 70%, 80% from here, around here. So I think the quality of the sequence is no problem. So let's try to 10 or 20 characteristics marked. So in my laboratory, just we try to mark here by giving the color like this. Okay, and from T to G here, let's try to search. There is some there's the same sequence or not. So we need to define in the case of this sequence data. Okay, let's try to search by using a tool of the word search. Yes, we found, yes. So as I said, this is the information from the forward primer, and this is information from reverse primers. But we carry that picture like this, and so there are some the overlap points, as I said. So it means around here, so the same sequence should be there. Yes, I got it already here. Okay, let's try to mark by giving color like this. Okay. So let's try to nearby sequence data, like before the colored area. So CAAA. Okay, let's try to check it. CAAA. Yes, that's right. So it means from here. So and after that around here, same sequences. So we can from this point we can combine the two sequence data like this. Okay, so let's try to after this area GGCC, right? Okay, GGCC, yes, same. Yes, we can successfully find the same sequence area, so it's okay to delete here. Okay, and so we can delete around here. Okay, yes, let's try to overlap this one and this one. Okay. Yes, so like this we can get almost the full length of 16 srDNA by using two primers and by combining the two sequence data from here to here. Yes. So if you want to know the sequence data, so longer the sequence data, this is just 1,600 base pairs, but for example 3,000, 5,000, so we need to use various primers, not 2 or 4 or 5, 6 primers, and we need to combine together. But if we increase the number of the primers, no problem. this is just the same way. By using Word, we can look for some the overlapped area like this and combine together, and so we can get the full length of the each genes. Okay. Okay, so from now I'd like to introduce to the how to construct the phylogenetic tree. Okay. Yes, so let's try to use our data of our students. Yes, my students try to isolate the lactic acid bacteria. Can you see here? Can you see here? Yes, here. So 1, 2, 3, 4, 5. Yeah, 5 strain we already isolated, and so we got the sequenced data like this. Okay. And so, when we construct the phylogenetic tree, we need to reference data. So it means the already identified species, and so already a sequence also was deposited on the website like NCBI. So from NCBI website, we can get the sequence data of 16 sRDNA of another lactic acid bacteria. Okay, so here, so we already, search the information of sequences about each lactic acid bacteria like this. Here, so Lactococcus lactis, and this one is one of the Lactococcus species, and then so like this. This one is the Lactobacillus plantarum, and another the species of lactic acid bacteria. Yes, yeah, Lactobacillus brevis and Pedicoccus. So like this, by the pre-screening these 5 strains, we can say that these are one of the lactic acid bacteria, but we need to identify as by comparison of sequence data. In that case, we need to correct the sequence data, the similar species, like as I said, the another kinds of the lactic acid bacteria. Yes. So here, so I use the software of the Memo notebook, or you can use a Word. But anyway, like this, one by one, against the one strains, we need to prepare the one sequence. Yes. And also, when we put this data in some software to construct our phylogenetic tree, so we need to set up this form. So it means that, as you can see, this symbol, and after that we need to put the name of the strain, something like that. Yeah, all data were set up like this. Yes, this symbol, and then after that the name or the bacteria or some strain number, and after that we can paste our sequence data. Yes, so this the format, we can say the FASTA format. So this is very important to utilize some software to for example, the edit, align, or construct the phylogenetic tree by using sequence data. Not only bacteria, but also all the organisms, the fish, mammalians. So could you please remember this the format, FASTA format, okay? Yes. So in the one file, I set up the sequence data like this, okay? Let's try to use this data. Okay, so firstly we need to carry out the alignment among the all sequence data alignment. So what kind of the software we can use? So in my case, yes, this one, the ClusterX. So this one is also one of the free software you can freely download in your computers and you can use. Okay, could you please remember ClusterX software? Okay, yes, could you please try to open that file? So you can select this one, load sequence. It means that already I showed this. The sequence data should be transferred into this software. Okay, let's try to load sequence. Okay, I found that data. This one, sequence. Let's try to open. Yes, already all sequence data was transferred into this software from isolate 5 string 1, 2, 3, 4, 5, maybe a very small characters difficult to see, but I can say 1, 2, 3, 4, 5. Okay, and so after that, different the sequence data are obtained from the website from in the NCBI. Let's try to move. Okay. Yeah, you can see 1, 2, 3, 4, 5, and the reference data like this, and so in each are strains, so we can already success to incorporate like this, okay. But at present, just the sequence data, just are paste in each line, but we need to align among the bacterial species. So in that case, we can put the button here. Do complete alignment. Yes. Let's try to do it. Okay, just okay. Yes, now alignment is is being carried out like this. Yes, calculating. So maybe actually it takes just 1 minute or something like that. Let's try to wait. Yes, I got the data like this. Yes, this is a result of alignment. As you can see, the among this all strains, for example, around here, yes, the same, the C, and all strains, this position, the the nucleotide should be, C. Yes, the next one is G, overlapped the same information, but for example, sometimes we can find the different among the species. For example, here T, but after that C, and T. So it means except for 1, 2, 3, 4, 5, 6 strains, or almost all the data shows T, but this difference should be depending on the bacterial species. Yes, like this, by alignment the among species in any information about the 16S DNA, we can find the difference, the position of the ATGC like this. Okay, so based on this data, so we can get the aligned sequence data set. Okay, I forgot one information about that. When we save this data after alignment, could you please select the method to save from here? Save sequence as. And so could you please the mark here, FASTA format. Yes, and okay. And so after the saving, we can get the specific data for construction of phylogenetic. Yes, could you please mark about the FASTA format? Okay. Okay, so to construct the phylogenetic tree, so we can use this one, Mega software, yes, this one, yes, Mega software. Okay, so this one also are the easily are obtained from the internet, because this is also the free software. Okay. Yes, so by using Mega software, could you please open the file? Yes. Yes, so the after alignment we can get of this software's sequences based on FASTA format. Could you please select the file? Yes, so the after using ClusterX and we did the alignment, we can get the various files like this one and this one. So we can see various kinds of files, but could you please use just only this type of the data? So you can see the mark of Mega software. Yes, could you please check this one? And so there are some questions. So just try to select the analyze, because already we finished align. Just we want to analyze, and we want to make the phylogenetic tree. Yes. So next question is what kind of the data? So nucleotide sequences. And okay. A protein coding nucleotide. Just you can set only yes. And there are some, we can customize the condition of the software, but you can select the standard. Okay, so already our data was the transfer from the cluster W to our mega software like this. Okay, so let's try to analyze and let's try to construct the phylogenetic tree, and please select the analysis here, and then so please select the phylogeny. Yes, and after that you can see there are some the commands, what kind of the tree you want to prepare. Yes, here, so our construct test maximum likelihood, something like that, and after that the neighbor joining. So due to your experiment, we can select the type of phylogenetic trees, but in our laboratory case, so simply we can select the neighbor joining tree, this one, second one. Okay, and please click it. Yes, so you want to use a currently activated, so it means our file, and so we want to construct our tree, so it's okay, just so you can click yes. Yes, so there are so many information about to construct the phylogenetic trees. Yes, so about the conditions, it is due to your experimental characteristics. But could you please remember for example this one, bootstrap method, and so replication. This is very important because this is very effect on the quality of the phylogenetic tree. So normally, so we can select the 1000, but if you select like a 100, so the data of quality of the data is not enough. Just please select the more than 1000. Okay, and so this is one of the statistic analysis. So we can freely select the how to analyze, how to carry out the statistic analysis. Yes, or what model you can select, but this one also depends on the kind of the samples. So could you please discuss with your supervisor and you can select it. So in my case, normally, like a cumulative parameter model or Tajima model, something like that, we can use. Okay, and so after that, if we put the compute, let's try to start to construct the phylogenetic tree. Yes, now making. Yes, I got the data. Yes. Okay, like this. Yeah, very quickly we can get the sequence, our phylogenetic tree. Yes, like this. Yes. So please look at here. Can you see it? Okay. Yes. So as you can see, there are some numbers. Yes. In our laboratory, I said 5D strains was isolated, and so strain 1 and 3 and 5 and 4. So could you please the position of cluster? Yes, these are Lactococcus lactis, and here is Lactococcus lactis, and subspecies Cremoris. Yes, so 4 strains 1, 3, 4, 5 was are located on these clusters. So it means that these strains are very similar to Lactococcus lactis species. Okay, but the case of strain 2 here, this one is similar with similar position. Yes, with the subspecies Cremoris. So it means in 5 strains, 4 strains is very similar species with Lactococcus lactis. subspecies Lactis, but a case of strain 2 similar with subspecies Culemoris. Yes, so there are so many another reference the lacticus bacteria like Lactococcus plantrum pedicocccus, but the cluster was significantly different with the our strain. So it means we can successfully identify our 5 strains. Four strains are Lactis. The one strain is the Culemoris. Okay. Yes. So like this, we can construct our phytogenic tree. So could you please utilize these the procedure in your experiment? Yes, maybe you are not, you will not use bacteria or microorganism, but as I said, our methodology was same with the case of another organism, plants, mammalians, fish. Okay, could you please remember how to use it? Okay, so after that, I will give the final mini report, and so could you please submit it to me, okay? Okay, so I introduced how to construct a phylogenetic tree. Okay, software of the BioEdit, Mega or ClusterX, something like that. So these software can be utilized in not only our laboratory but also in another field. Yes, could you please remember how to use this software? Okay. Yes, so I will give the mini report related to about the phylogenetic tree construction. So that's why as a shortly I made the movie to keep time for you to prepare the mini report. So meaning the to prepare the phylogenetic trees. Okay, so the detail of information about the mini report, could you please download attached files, and so could you read it, and so please submit the result to me. Okay. Okay, yes, so my class about the Biotechnology II are finished. Okay, yes, so maybe you will have the test for graduation, to test about the semester test. Okay, yeah, so could you please keep your health, and so maybe I think this year 2021 still there are some problems about their COVID-19, so we cannot estimate when we can dissolve these problems, and so how we can dissolve these problems. But anyway, no way. Yes. So to keep our life, to keep our health, so we need to carry out the experiment, research, and graduate. Okay. Yes. Okay, so if we have some question about the mini report or anything else, so could you please contact with me by sending email. Okay, bye bye, see you later and elsewhere. Okay, bye bye.